This is Chapter 6, the Skeletal System, Bone Tissue, Part 2. We were talking in the last video about bone formation called ossification or osteogenesis. And we had said that bone formation always involves the replacement of a pre-existing connective tissue. Osteogenesis occurs by two different methods during embryonic development. So one method is intramembranous ossification, which produces spongy bone, and this bone may later be remodeled to form compact bone. And endochondral, and sometimes you see this as endochondrial ossification, is a process by which cartilage is replaced by bone. So intramembranous, within a membrane, endochondrial, within cartilage. And endochondrial forms both compact and spongy bone. So intramembranous ossification occurs within a membrane, and it's used in forming the flat bones of the skull, mandible, and clavicle. At about the 5th week of embryonic development, a mesenchyme membrane forms around the developing brain. The bone forms directly within this membrane from mesenchyme cells. Recall from Chapter 4 that mesenchyme is the embryonic connective tissue from which all other connective tissues develop. So in this method, bone replaces mesenchyme. So in intramembranous ossification, and remember, bone here replaces a membrane. The first stage is the development of ossification centers. So the first thing that happens is that mesenchyme cells cluster together and differentiate into osteogenic, also called osteoprogenitor cells, then into osteoblasts, which secrete bone matrix. These clusters of osteoblasts are called ossification centers. So ossification centers is basically are where these the bone formation begins and spreads out from there. Then secondly, the second step is calcification. Once surrounded by a matrix, osteoblasts become osteocytes, which then secrete mineral salts into the matrix to harden it up. 3. Trabeculae form. Now, trabeculae form spongy bone. So cells in the spaces of trabeculae form the red marrow. And then your periosteum forms. Osteoblasts and mesenchyme at the periphery form the periosteum. Spongy bone on the surface is later replaced by compact bone. Everybody is aware of fontanelles or soft spots in the baby's skull at birth, and these soft spots represent large membrane spaces where ossification is not yet complete. It takes about 2 years for it to be complete. So in the center of every flat skull bone, you will have bone formation occurring, and then it spreads out from the center. And this is happening in each of the skull bones. Much of the new bone is then remodeled and reshaped as the bone is transformed into its adult size and shape. This figure here is from your book, and it demonstrates the stages. Here we can see some of these flat bones forming by intramembranous ossification. So first of all, somewhere in the membrane we will have some of the mesenchyme cells differentiating into osteoblasts, which start secreting bone matrix. Okay, the organic portion of the bone matrix. Then we get calcification. Once these osteoblasts are completely surrounded by matrix, we then call them osteocytes. We still have osteoblasts on that periphery here, but these cells are osteocytes. And they start secreting calcium salts into the matrix to harden it up. This step is calcification. Third, then we have the formation of trabeculae. An extracellular matrix develops into trabeculae that fuse to form spongy bone. And then, lastly, we have the development of the periosteum. Mesenchyme cells on the periphery here differentiated into osteoblasts, and they are forming the periosteum. So you can see what you have at the end of intramembranous ossification. Kind of looks like a bone sandwich with the interior of spongy bone, but the bone just under the periosteum has been remodeled into compact bone, so it's kind of like a bone sandwich with the interior spongy and the outer parts of our sandwich are compact bone. Now, the other method, endochondral ossification, is the method used in the formation of most bones of the body, especially the long bones, and it involves replacement of hyaline cartilage by bone. Endochondrial means within cartilage. So cartilage models are formed first and then replaced by bone. And most of the fetal skeleton is first laid down in hyaline cartilage to be later replaced by that bone. There's one primary center of ossification in the diaphysis and two secondary centers, one in each of the epiphyses. So the first thing that happens is we have the development of a hyaline cartilage model, where mesenchyme cells develop into chondroblasts, not into osteoblasts this time, but into chondroblasts, which are going to secrete cartilage matrix. And the model is surrounded by a membrane called the perichondrium. Remember that it was periosteum around bone. If the membrane surrounds cartilage, it's perichondrium. So we develop this cartilage model, and then secondly, we need growth of this cartilage model. Appositional growth is growth in width. Chondroblasts in the perichondrium add more matrix to the periphery of our cartilage model. Interstitial growth is growth in length. Chondrocytes divide within their lacunae and add more matrix between the cells. Now, bone cells cannot add more matrix, or osteocytes cannot add more matrix. Once they're completely surrounded by matrix, they can't add any more. But because the extracellular matrix of cartilage is somewhat rubbery and stretchy, the chondrocytes can actually add more matrix from within. Chondrocytes in the center of our cartilage model hypertrophy or enlarge and release calcium. Then this cartilage in this area of the cartilage model becomes calcified, so we get calcified cartilage. Then we have the development of a primary center of ossification. We have our cartilage model, now we need to ossify it. What happens here is a nutrient artery penetrates the perichondrium into the center of the cartilage model. When the nutrient artery penetrates into the center of the cartilage model, it stimulates osteogenic cells in the perichondrium to differentiate into osteoblasts, which secrete bone matrix. The membrane in this area is now called the periosteum because it's surrounding bone. Periosteal capillaries penetrate the calcified cartilage, inducing the growth of spongy bone. This happens at the primary ossification center. Then ossification spreads towards both ends of our cartilage model. Number 4, development of the medullary or marrow cavity. Osteoclasts entering the model with the nutrient artery breaks down some of the newly formed trabeculae in the center of the model, leaving a medullary or marrow cavity in the diaphysis. Then we have ossified our diaphysis. Now we need to ossify the epiphyses. So we need to develop secondary centers of ossification. This occurs when branches of the epiphyseal artery enter the epiphyses and secondary centers of ossification form in each of the epiphyses. Spongy bone is formed, but here no medullary cavity is formed. And lastly, formation of articular cartilage and the epiphyseal plate. The original cartilage model is almost completely ossified or replaced by bone, except for the articular cartilage, which remains throughout life, and the epiphyseal or growth plate, which is responsible for growth and length, but does become ossified in adults. Now let's look at the pictorial description of this process. Maybe it's a little bit clearer. Our first stage was the development of a cartilage model. Remember here, mesenchyme cells differentiate into chondroblasts, which form cartilage matrix. So we have a cartilage model being formed. Secondly, we have the growth of the cartilage model, and the chondrocytes within their lacunae will undergo cell division and produce more chondrocytes as well as more bone matrix, so the whole model grows. Now what's happening here is you can see in the center of this model, the cartilage cells here have hypertrophied or become enlarged, and they burst, releasing calcium into the matrix, and then you get a little area of calcified cartilage. Next, we have the development of the primary ossification center. Here we can see the nutrient artery penetrating into the center of our cartilage model. Going along with this nutrient artery, we have osteoblasts and osteoclasts. The osteoblasts are going to lay down bone matrix, spongy bone, in the center of our diaphysis. Now we get this bony color, and you can see that in this area here, just where bone is forming, now that membrane is called the periosteum. Elsewhere, it is still the perichondrium. So in the last step, we had osteoblasts entering into the center of our cartilage model, replacing the center with bone. But osteoclasts also traveled in with these blood vessels. So whereas the osteoblasts lay down spongy bone, the osteoclasts are now sculpting out the center of this model here, so that we get the formation of a marrow cavity. So first we lay down bone matrix, and then we carve it out or sculpt it out with the osteoclasts. Next, development of the secondary centers of ossification that have to occur in each of the epiphyses. Here you can see arteries, epiphyseal arteries penetrating into the epiphyseal end of our cartilage model. With these blood vessels come osteoblasts. They start laying down bone matrix. We're forming spongy bone. And lastly, the formation of articular cartilage and the epiphyseal plate. You can see that in the epiphysis, not all of the hyaline cartilage is replaced by spongy bone. The outer surface here remains hyaline cartilage. This becomes the articular cartilage, and a little sandwich of hyaline cartilage remains here to become the epiphyseal or growth plate. Growth and length of the diaphysis occurs only in this little cartilage growth plate. Ossification contributing to bone length is by interstitial growth of cartilage at the growth plate, and it's usually complete by about the 18 to 21 years of age. So bones can continue to lengthen as long as that growth plate is active, but it does begin to ossify in the late teen years. Bones can still continue to thicken or grow in width by appositional growth, under the periosteum, just throughout life. And these the osteoblasts in the periosteum are capable of repair of bone even after the epiphyseal growth plates have closed. So bone growth in length is by interstitial growth of the cartilage at the growth plate. Growth in width is by appositional growth under the periosteum, and that can occur throughout lifetime. And I always remember appositional growth, growth in width or thickness, because if I was making a clay model of a bone, the way that I would thicken it up is to apply more clay around the periphery to thicken it up. In the figure that you see down here, you can see here's the femur, here's the tibia of a 3-year-old, so you would expect to see the cartilage growth plates here and here. And again, it is within these growth plates that is the only place where bone can grow in length. The cartilage grows here and then is ossified, and that lengthens the diaphysis. The next slide is going to show a magnified view of the cartilage in the epiphyseal plate. The cartilage in the growth plate is producing new cartilage by interstitial growth in such a way that it grows away from the center of the diaphysis, and it elongates the diaphysis at each end of the bone. The cartilage nearest the diaphysis becomes ossified. This continues until the hormones of puberty, like estrogen and testosterone, which hasten not only growth at the growth plate, but also its ossification. So growth and length continues until these hormones of puberty have result in the complete ossification of the epiphyseal plate. Once the epiphyseal plate is completely ossified, it becomes known as the epiphyseal line, and no more growth and length is possible. Here is a large magnified view of the epiphyseal plate. It's all cartilage here. Here is the diaphyseal side, the epiphyseal side. So our bone is growing in this direction. This is the oldest cartilage up here. Here's the newest. And you can see here, here's a zone of proliferating cartilage, hypertrophic cartilage. So here's actively forming cartilage. This is older cartilage, which is now becoming calcified, and this is even older yet. On the diaphyseal side, this cartilage has become ossified. So this growth plate continues to march outward from the center of the diaphysis to elongate that diaphysis. And here you get yet another diagram to show this. Here is your epiphyseal plate, and you can see the cartilage here is going to grow in this direction. And as the cartilage grows in this direction, ossification occurs behind it. Once again, you have an elongation of that diaphysis. Now, the following slide demonstrates growth in width by appositional growth under the periosteum. The sequence of illustrations shows that new bone growth under the periosteum is occurring at such a rapid rate that it forms ripples on the surface of the bone. Blood vessels lying in the grooves of these ripples eventually become trapped by matrix. The periosteum that was trapped along with the vessel now becomes known as endosteum, because now it's inside the bone, not outside. Concentric lamellae will be laid down around the new central canal, and the result is the formation of a new osteon. So here we see the phases. Here's your periosteum and some periosteal arteries lying there. Here we have basically some circumferential lamellae. In the periosteum, we know that there are osteoblasts. So the osteoblasts here are going to start laying down bone matrix so fast that the surface of the bone ripples, and you can see as the ripples become more prominent, this blood vessel lying in the groove of one of the ripples will eventually become entrapped. So here we can see that trapped blood vessel, and now the periosteum here is the endosteum. Remember that there are osteoblasts inside here, and they're going to lay down bone matrix in the only direction they can, which is inward. And so they're going to form rings of hard matrix going inward until you get the formation of a central canal. So even though it kind of looks like the cross-section through a tree adds its layers from inside out. Here, these lamellae were laid down from outside in until they just ran out of room. So here is our central canal and our newly formed osteon. And we can see that as this process continues, this blood vessel is about to get trapped, so that's going to become the center of a new osteon. Important to note, the only way bone can grow is by appositional growth in width by osteoblasts in the perichondrium. Bone tissue cannot grow from within by interstitial growth. Bones can grow in length only by interstitial growth in cartilage at the epiphyseal plate. Cartilage can lay down new matrix by both interstitial and appositional growth. So cartilage can grow from within and also on the edges. Bone can only grow on the edges. Bone growth and remodeling. You always have to have a balance between the actions of osteoclasts and osteoblasts. Bone remodeling does require the action of both osteoblasts that deposit new bone and osteoclasts that break down old bone. The whole process is not completely understood, but it is an ongoing process throughout your life. I think I may have mentioned at the beginning of this lecture that your skeleton is estimated to replace itself completely about 7 times during your lifetime. The breakdown of bone is called resorption, and we know that the formation of new bone is osteogenesis or ossification. So bone remodeling involves osteoblasts and osteoclasts. There are factors that affect bone growth like hormones, human growth hormone, thyroid hormone. Hormones like this increase general tissue growth of all tissues in the body. Sex hormones at puberty, estrogen, testosterone, hormones like that, stimulate bone growth, but they also stimulate ossification of the growth plate, causing growth and length to cease. So we know that in the teen years especially, there's big spurts of growth, and this is due to puberty hormones, so causing large amounts of growth. But at the same time, the growth plates are ossifying, and once those growth plates have become completely ossified, no more growth and length is possible. Nutrition. You have to have an adequate supply of vitamins and minerals, especially calcium and phosphorus. Also, vitamin A stimulates osteoblasts. Vitamin C is necessary for collagen formation. Vitamin D is necessary for calcium absorption. Vitamins K and vitamin B12 are necessary for the synthesis of bone proteins. So you can see you need the right things in your diet in order to be able to form bone. And of course, genetics determines the potential final size of bones. You may have the genes to be very, very tall, but if your nutrition is poor, you won't realize that genetic potential. However, if you're in a situation where you have good nutrition, you will realize that genetic potential. If you have good nutrition and the genes for being short, then no matter how good that nutrition is, you're going to be short. So it is your genes that determine the potential final size, but it's some of the other factors that decide whether that will be in fact realized. Bone is a major storage site for calcium. About 99% of your body's calcium is stored in bone. It's stored in bone by osteoblasts when they make bone matrix, and it's released by osteoclasts when they break down bone matrix. When blood levels of calcium fall too low, osteoclast activity increases. When blood calcium levels are too high, osteoblast activity increases. Calcium homeostasis primarily involves the action of hormones. 2 hormones in particular. Parathyroid hormone is one of these. It is secreted by the parathyroid gland, which is on the dorsal side of the thyroid gland. This gland stimulates this hormone that stimulates osteoclast activity and raises blood calcium levels. Also, you get a loss of calcium in the urine. So parathyroid hormone is going to work on osteoclasts, and it's also going to work on the kidney to decrease the loss of calcium in the urine, and also along with calcitriol, which is active vitamin D, the hormone active vitamin D. These are going to increase the absorption of calcium from the intestines, from your diet. So parathyroid hormone stimulates osteoclasts to break down bone, and this releases calcium into the bloodstream, and it also decreases the loss of calcium in the urine and helps to increase the absorption of calcium from your diet. Calcitonin is released by the thyroid gland in response to high calcium in the blood, and this stimulates osteoblast activity to lower blood calcium level. So when osteoblasts are more active, they are making bone matrix, and this, of course, requires calcium. So this is going to increase the deposit of calcium in the bone. So you get it out of blood and deposit it in the bone. So these 2 hormones, parathyroid hormone and calcitonin, have opposite actions. Let's talk for a moment about fracture and repair. And no matter how bone is fractured, and there are many different ways to break the bone, repair proceeds in a predictable pattern. The first step, which occurs 6 to 8 hours after injury, is the formation of a fracture hematoma or blood clot as a result of blood vessels breaking in the periosteum and in osteons. So you can see the broken bones here and the broken blood vessels, and you can see a blood clot forming. The second step is called the formation of a cartilage callus. This blood clot is replaced by a fibrocartilaginous callus. Phagocytes are going to remove cellular debris, and fibroblasts are going to start depositing collagen to form this callus. Okay, so now we have a cartilaginous callus. Now we can ossify that cartilage. So the third step is callus ossification. The cartilage callus is replaced by a bony callus. Osteoblasts form a bony callus of spongy bone. So you can see the spongy bone forming here. All these little cells here represent osteoblasts. So you can see they're forming bone, but the first kind of bone formed is spongy. And steps 2 and 3 can take weeks or months. It just depends on which bone it is and how much weight that bone bears. And lastly, we have remodeling, because remember, we formed spongy bone here, but we don't want spongy bone on the exterior surface of our bone. So the spongy bone is then remodeled into compact bone. We know that remodeling includes both osteoblasts and osteoclasts. Now the fracture line disappears, but evidence of the break usually remains. Exercise places stress on bones. Bone tissue becomes stronger through deposition of mineral salts and production of collagen fibers by osteoblasts. Unstressed bones become weaker. For example, astronauts in space do suffer a rapid loss of bone density. And so what's really happening here is you have the main mechanical stresses on bone resulting from the pull of skeletal muscles on that bone, and also the pull of gravity in weight-bearing activities. As the muscle mass increases, the bone mass has to increase to support it. Exercise may also stimulate the release of calcitonin, which promotes bone formation. In aging, there's a decrease in bone mass that occurs as the level of sex hormones diminish during middle age. This happens especially in women after menopause. Bone resorption, or bone breakdown, by osteoclasts starts to outpace bone deposition by osteoblasts, so you're breaking down bone faster than you're making it. Since female bones are generally smaller and less massive than males to begin with, old age has a greater adverse effect in females. And also, the loss of bone mass in females begins after the age of 30, and in males it doesn't start till after the age of 60. So we have these very small little trabeculae that are getting thinner and thinner, and eventually you're going to get compression fractures as the thin trabeculae just cannot support the weight. So there are two principal effects of aging on bone tissue. Loss of bone mass, the loss of calcium from bones, is one of the symptoms in osteoporosis, and also brittleness. Collagen fibers give bone its tensile strength. Protein synthesis decreases with age, so you're losing a lot of collagen. The loss of the tensile strength by collagen causes the bones to become very brittle and very susceptible to fracture. And here you can see two pictures side by side. The one on the left here is normal bone, and what you're looking at is the trabeculae of spongy bone. And then here is osteoporotic bone, and you can see it looks very porous. These trabeculae are being broken down by increased osteoclast activity and perhaps lowered calcium in the diet, and these trabeculae are not going to be able to support much weight. And that is the end of part 2 of chapter 6.